So in the first part of the video, I went over my expectations for the course, and we looked at mechanisms for evolution. And one major confusion for people was this idea of founder's effect. And people asked me about a human example, and this is actually an example of Amish settlers that settled in certain parts of Pennsylvania. And to this day, you'll still see some of these Amish communities have a lot more people with 6 fingers versus 5 fingers. So this is mechanisms for evolution. The other confusion a lot of people asked me about were about speciation. So let's move on to speciation. Speciation is the evolution of a new species. To have speciation, you have to have some sort of reproductive barrier, because you want to restrict gene flow. Okay. Now, to restrict gene flow, okay, you have 2 major options. Okay, you can create an actual geographic separation, or you can create this reproductive barrier without a geographic separation. So the first dividing line here is to geographically isolate a population. Okay, that type of speciation happens to be called allopatric speciation. In allopatric speciation, the reproductive barrier is geographic isolation, like the squirrels on either side of the Grand Canyon. Okay? And this geographic isolation is a type of pre-zygotic barrier because it's before the egg and the sperm get together. The other type of speciation is called sympatric speciation, and the reproductive barrier is not geographic. So the reproductive barrier can be temporal, like they reproduce at different times, behavioral, mechanical, etc. These are all examples of pre-zygotic barriers because they occur before the egg and the sperm get together. So we're stopping gene flow so that enough variation and mutations can build up in the separated populations to possibly cause speciation. Now, sometimes groups that have been separated come back together and can still reproduce, but you have problems with the hybrid, which means you have problems with the offspring from the 2 different groups. Okay? When this happens, these are called post-zygotic barriers because they are after the sperm and the egg have united. Okay? So in speciation, it's not like overnight snap your fingers, you have a new species. Although that can happen in plants sometimes when polyploidy occurs, when their chromosome number overnight duplicates and then they're automatically a new species. But in a lot of animals, this takes a little bit more time, right? So you have like a big population of one species, okay? And then for some reason, they're reproductively separated. So here now is population 1, and here is population 2. So they are separated. This barrier could be geographic. It could be like the Grand Canyon separating them. It could be a different type of reproductive barrier, like behavioral. They don't do the same mating call. Okay? And so this goes on for many generations, and this population starts to change. But let's say, for some reason, the populations, a couple individuals from this population, many generations down the line, meet up with a couple individuals from this population. And they mate, and they have an offspring. Okay? These barriers separating them are pre-zygotic because they are preventing the sperm and the egg from uniting. Okay? maybe when these two meet up again, there's another prezygotic barrier. Like they have, I don't know, their shells don't fit together if they're insects or something. Whatever. So prezygotic just means the sperm and the egg can't unite. But let's say for some reason these two get together and they're able to mate and produce an offspring. This offspring is the hybrid. Okay? If they're still able to produce a hybrid and their children are able to produce children, they're still part of the same species. But if this hybrid is weak or is sterile or the children of this hybrid are weak, those are problems with the hybrid. Those are postzygotic problems, and now they're part of a new species. So if at any point when organisms from two populations can no longer mate and produce fertile offspring, then speciation can occur, right? Allopatric speciation is when the reproductive barrier is geographic. Sympatric speciation is when organisms are still living close to each other, but they're separated for other reasons. Prezygotic barriers are barriers to the sperm and the egg uniting. Postzygotic barriers are the sperm and the egg united, however, the offspring is not viable or is sterile, etc. So those are speciation questions answered based on what you wrote on the form. So I've gone over your speciation questions, and I want to preview the lactase activity, which is going to be your homework. It covers mutations, pedigrees, and natural selection. So each student will get a copy, a Google Doc of this lactase persistence evidence for selection. This is a really, really interesting topic, and it's about lactose intolerance, which either you might be lactose intolerant or you probably know something that's lactose intolerant. And it looks at the evolution of lactose tolerance and lactose intolerance. So you'll watch this HHMI video, and then you'll answer these questions. And then you'll look at this figure about global distribution of lactase persistence, and you'll answer these questions. And then you'll look at this diagram of evidence of selection and answer these questions, and then you'll submit it. Now, the purpose of this assignment is to, one, keep your motivation up. It's super interesting and something that you guys, it probably either affects you or you know somebody that it affects. Second, it's a great review of selection. And third, it really hits home some of these things you need to review for the AP exam, including diagram analysis, including data analysis, including pedigree review. So these are some really important skills for the AP exam and for life, how you analyze diagrams. So we're kind of hitting at this idea of we're reviewing evolution, we're doing something that's interesting, and we're reviewing for the AP exam. So this assignment, it will be due next week. Also, I'm going to ask you to work on AP Review Guide 3, which will be posted as well. So by next week, you'll have the lactase activity due, and you'll have AP Review Guide 3 due. And then on Monday, I will post the videos for our next topic on cladograms, which we'll watch by class on Thursday. So we've talked about speciation, we've previewed the lactase activity. Let's talk about the AP exam. We're working on AP review guides, and I'm going to give you an update on the test, and we're going to practice reading a graph and doing scientific practice. You guys can all be successful on the AP exam. Please reach out to me via email if you're not sure whether or not you want to take the AP exam. I think you guys can be very, very successful at it, regardless of whether or not you think you'll use it in college. Most colleges are accepting it. It's just great practice. So please let me know what you're thinking. So what will the AP test look like? It will be a 45-minute test that you can take on your computer. Next week I'll look at the tech with you, what tech you'll need. And it's only 2 questions. They're both FRQ questions. The first question will be 30 minutes, okay? And it is a free response question. And this is what it will look like. It will be interpreting and evaluating experimental results. So just like in that lactase assignment, okay, you'll have a scenario, like the lactase scenario, accompanied by a data table and a graph. You won't have to graph, but you will have to analyze graphs. And part A will be 1 to 2 points. You'll have to describe or explain the biological concept or model in the diagrams that they give you. Part B, You'll have to identify experimental design procedures, independent variable, dependent variable, controls. So we'll practice that as well. Part C, you'll have to analyze data. And part D, you're going to have to make and justify predictions. So a lot of the stuff we're going to have you practice is on claim, evidence, and reasoning, because this is a big part of question 1 on the AP exam. And if you go into science, computer science, engineering, etc., these are skills that you'll need in your life. So we're practicing life skills and AP exam skills. That will be the first question on the online AP test. The second question will be a conceptual analysis 4-point question. And this will be, I think, a 10-minute question. You'll have to describe a biological concept or process, explain it, predict changes, justify predictions. Again, claim, evidence, reasoning is huge here. So we're going to end today with a practice question from 2018. I suggest at this point you pause the video, read the question, and then you'll have a better idea of what I'm talking about. So this question looks at bed bugs, okay? And the first thing I would do is I would read the figure if I was doing this problem, and I would look at the graph and look at the data table, and then I would read the question. So make sure you've done that before we move on. So let's look at this together. This says percent survival of 5 strains of bed bugs. So these 1, 2, 3, 4, and 5 are 5 different types of bed bugs. A plus indicates that they have each of these genes. A minus indicates that the gene was deleted. So this first strain of bed bug had all 3 of the genes that we're looking at. The second one had P450, that gene was deleted, but had APC8 and CPS, and so on. So basically, once you read the question, make sure you've read the question, it says, hey, scientists are worried because bed bugs are resistant to a chemical that kills bed bugs. Okay, and they think that one of these 3 genes, P450, APC8, or CPS, is helping cause that resistance, is helping those bed bugs survive. Okay, so they took a normal bed bug strain, and then they deleted different genes in each of these strains. So in strain 2, they deleted this gene, but kept these 2. In strain 3, they deleted this gene. In strain 4, they deleted this gene. In strain 5, they deleted 2 of the 3 genes. Okay, and then they saw how well the bed bugs survived. These bars, okay, represent error bars. These represent the range in which we're 95% confident in our answers. So while this is the mean, this is the range of where our mean could have been. Here's the big thing. If the error bars overlap, there's no significant difference between different bars where the error bars overlap. So for instance, in strain 1 and strain 2, here's the error bar in strain 1, saying our mean could have been anywhere within this error bar. Here's the error bar with strain 2, our mean could have been anywhere here. And you see, look, strain 1 and 2, the error bars don't overlap. So there's a significant difference in the percent survival rate for strain 1 bed bugs and the strain 2 bed bugs. Okay? In contrast, look at how big this error bar is for strain 4. It overlaps with strain 3 and strain 2, and actually strain 1. So there's no significant difference between strain 4 and strains 1, 2, and 3. Okay? So that's what the error bars mean. So part A says identify the control strain in the experiment. Okay? Use the means and confidence intervals in figure 1 to justify the claim that APC8 is effective at providing resistance to beta, cyfluorethane. Okay, so the control strain is the one used for comparison. So I would pick strain 1 as the control strain because all 3 of the genes are activated, just like in the normal bed bugs. Okay? Use the means and confidence intervals in figure 1 to justify the claim that APC8 is effective. So at providing resistance. So when APC8 is active, you can see the means are much higher, whereas when it's inactive, the means are much lower. Okay? My only difficulty, and actually, I have a small problem with that because as you can see, in 3, okay, where APC8 is inactive, the mean is lower. However, the error bar does overlap with 2. So it actually seems that both APC8 and P450 both really impact the survival rate of the bed bugs. When they're active, the survival rate is higher. when they're inactive, the survival rate is lower. And you can actually see that in strain 5, where P450 and ABC8 are both inactive, and you can see the mean is much lower, and actually I don't think the error bars overlap with any of the other error bars, so there is a statistically significant difference. Okay. What I want you to do before next class is write out your answer to A, write out your answer to B, just jot it down on a piece of paper, and we'll go over it together next class. Thanks, guys.